Welcome, everyone. I want to welcome you to the Protecting Southeastern Freshwater Biodiversity webinar. I'm April Engel, and I'm the Science and Policy Associate at River Network, and we're excited to have you join us today and for you to learn more about this study to develop regional conservation priorities and identify needed management and policy actions related to Southeastern freshwater biodiversity. Before we begin, I want to just share with you a few logistical reminders. We're recording today's webinar, and we will email you a link to the recording tomorrow. All participants will be muted throughout the webinar. However, you should see an orange button with a right pointing arrow in the top right of your screen, and pressing that button will give you access to a control panel where you can access the questions box in a pane that says questions. And that's where you can ask questions anytime throughout the webinar. And also, we have a webinar evaluation survey set up, and that's going to open on your computer when the webinar ends and will be included in the follow-up email. So please fill that out for us because your feedback makes a really big difference for us. So, and thank you in advance for doing that. So to kick us off, I want to start out by briefly just telling you a little bit about River Network. So for 29 years, we've worked to support the 2,000 plus river and watershed organizations that work at the local and state levels around the country, while also striving to engage a much broader community to support healthy rivers and water resources. Through our website and our annual River Rally Conference, our publications, through convenings and networks of peers and experts, as well as resources and direct consultation with our team here at River Network, we work to support the individuals and groups and coalitions who are working to protect and restore their waters across the United States. And our team here at River Network includes people who have experience and understand the challenges of working on the ground to take care of local waterways, as well as the importance of science to making sound decisions and how to use policy and advocacy to create lasting change. And you have the opportunity actually to join River Network if you haven't already, and we have both free and paid options. And when you or your organization join River Network as dues paying members, you become part of a community where you can gain access to premium benefits and resources, including an online network of both peers and experts, as well as discounts on our annual River Rally Conference, really significant discounts, and other trainings, as well as discounts on equipment and publications. And we'll also help promote your job openings, your events, and your work to a national audience through our website, our monthly e-newsletter, and our social media. So when you go onto our website, you can check out the professional, student, and organizational member options on our website and decide which one is best for you. So moving on to today's webinar, it's being organized as part of the Southeast River Flows Peer Learning Network. And in 2007, River Network, myself along with my colleague Catherine Baer, who is on the webinar with us today, we worked alongside a steering committee of NGOs who are working to protect and restore Southeast river flows to launch what we're calling the Southeast River Flows Peer Learning Network. And basically what that network is a platform where the network of community-based river protection NGOs from throughout the Southeast who are working on protecting and restoring the river flows can learn from each other and other experts, leverage their skills and efforts, create new alliances, and spark innovative and collaborative approaches, and work together to break through the barriers encountered in protecting river flows. And as I'm sure all of you are aware, there are many reasons that we want to protect and restore Southeastern rivers. We rely on them for drinking water, as places to swim, fish, and boat, to nourish our coastal bays and estuaries, as places of cultural and spiritual significance, to produce food and goods and energy. And as we'll learn today, the Southeastern United States is a global hotspot of freshwater biodiversity. So today's webinar is going to focus on the biodiversity of the Southeast rivers by showcasing the Southeastern Freshwater Biodiversity Conservation Strategy, And it's a data-driven analysis to rank southeastern watersheds based on their aquatic biodiversity, endemism, and imperilment, conducted by the staff of the River Basin Center at the Odom School of Ecology at the University of Georgia and the Tennessee Aquarium Conservation Institute. Today's presenters are Dr. Duncan Elkins and Dr. Bernard Kuhaida. And Dr. Elkins is a postdoctoral associate at the River Basin Center at the Odom School of Ecology at the University of Georgia and is the project manager of the Southeastern Freshwater Biodiversity Conservation Strategy. And Dr. Kuhaida is the science program manager at the Tennessee Aquarium Conservation Institute, and he's an expert in surveying and monitoring threatened and endangered species from aquatic systems in the Southeast, in part to help evaluate the effectiveness of conservation programs. So before I turn it over to Duncan and Bernie, and before we begin, we want to just learn a little bit about you. So I'm going to launch a few polls, and we'd love for you to weigh in. So what I'm going to do is I'm going to launch the first of these polls, and what we'd like to know is where do you work or where do you volunteer? And you can select all that apply. And I'll give you a few minutes to take the poll, and then I will share the results. All right. And it looks like 100% of you are working in the Southeast United States. So great, thank you for sharing that information. All right, we have one more poll that we'd like to share. And we'd like to know, what kind of organization do you work or volunteer for? All right, it looks like most of you have voted, and it looks like we have about 85% of you work for conservation or environmental organizations, 15% for academic institutions, and 15% for government. Sorry, I needed to share that with y'all there so you can see it. And great, so what I'm going to do now is turn it over to Duncan and Bernie, and if you'll give us just a few minutes to bring their slides up, they will get started.
Okay, thanks, April. Can everybody see that?
Yes.
Okay. Again, thanks, April and KB for giving us an opportunity to present as part of this seminar series and talk a little bit about this work that we here at the River Basin Center started with the folks at Tennessee about this time, actually, in 2015. We officially wrapped up the first version of this prioritization around December of 2017 or 16, sorry, and we have been working since then to revise it and to work on a couple of projects that are outgrowths of the prioritization here that we'll try to talk to you about at the end in the sort of next steps section of the presentation. So we have known for, well, ever since computer graphics were not very good at all, that the Southeast was a real hotspot for aquatic biodiversity. This is a paper from 2008. This was not the first paper to observe that certainly in the temperate world, the southeastern US is the real locus for fish diversity. The map for amphibians and reptiles is pretty similar. Maps for mussels look a lot like this as well. And you know, fast forward to 2015, the computer graphics have gotten a little better, but the picture hasn't changed much. This was a paper that came out just as we were starting our project in the Proceedings of National Academy of Science, where they looked at fish and a variety of other taxonomic groups to come up with a new set of conservation priorities for the lower 48. And you can see that this little band across North Georgia, northern Alabama, and the northeast corner of Mississippi, encompassing most of Middle Tennessee, is a real hotspot for fish diversity in the continental U.S. April, I think we talked about doing another slide or another poll right now.
Yes, I am going to launch that poll right now,
Okay.
Duncan.
Maybe we're kind of preaching to the choir if everybody in the call is affiliated with a Southeastern project.
Probably,
Yeah.
but that's okay.
We just wanted to sort of see if you knew or you wanted to take a guess at how much of the United States aquatic biodiversity is concentrated in the United States. And it looks like we've had most people take the poll. So let's close it and share it. And it looks like people's educated or not, or guesses are that between that 19% think it's between 25 and 50%. 25% of you think it's between 50 and 75%, and 56% of you think it's more than 75%.
And I would say that depending on how you slice it, the number is definitely one of those final two categories. What would you say, Bernie?
Yeah, I mean, for something like freshwater mussels and salamanders, it's well over 75%. I know for fishes, just in 3 main watersheds in the Southeast, it's half the diversity of the United States and Canada. So yeah, it's approaching 75, if not over, definitely. And then, just not to bore you with a bunch of maps, we actually have some pictures of the actual critters that we all care about. And since I've done a lot of work in Alabama and I had those numbers readily available, just to give you an idea, Alabama leads the country in the number of species of freshwater turtles at 25. Likewise for crayfish, I think is next. The number there is 97, and that's growing all the time with new descriptions. Moving on to mussels, it's about 182 species, and we break the 200 mark with snails, which are pretty fascinating animals. 203. The taxonomy is a little crazy with these, and genetics are almost as crazy, but people are working through it. And then, of course, the best group, since I am an ichthyologist, are the fishes. And we've got about 325 species of freshwater fishes in Alabama. So that just gives you an idea of some of the incredible aquatic biodiversity that there is in the Southeast.
Right. And so, as I say, this was not a new observation in 2015 that the Southeast has exceptional aquatic biodiversity. But as we were reviewing that literature, we realized that previous prioritizations didn't always use the best biological data sets. They either were sort of global synthesis papers where they really didn't have time to dig into the local data at all, or things like that Jenkins paper use data from NatureServe, which is great for some areas and some species, and perhaps not unassailable for other taxonomic groups in other areas. And so we thought that being fish people and having connections deep into the crayfish and mussel community, that we could put together a real deep dive into where aquatic biodiversity at least has historically been recorded in the Southeast. It's not a knock on those previous prioritizations to say that they didn't use the best biological data sets, because as you probably have already gathered, that's really a moving target. Bernie put together this slide on sort of new descriptions of fishes in the Southeast. You want to talk again?
Sure. So there's going to be a series of slides here, mostly of fishes, but also mussel, snails, and crayfishes of new species that have been described in the Southeast here and outside of the Southeast in the last just 13 years. Back about 8 years ago, the president's darters were described. Just about 4 years ago, the Caney Fork darter was described here in central Tennessee. We had several different sculpin described about 8 years ago. New snub-nosed darter, the bankhead and muscadine darter are brand new. Alabama bass was elevated from the spotted bass there in the Mobile Basin. Sticking with the game fishes, there's been a explosion of different species, which used to be the red-eye bass, Micropterus coosa. That's just now in the Coosa River system. And in the Mobile Basin, just about every river system above the fall line, the Tallapoosa, Cahaba, and Black Warrior have a unique form, now species of red-eye bass, Chattahoochee, and then there's still an undescribed one over in the Savannah. Moving on to other taxa, we've got the cypress floater, named after Paul Hartfield, a Fish and Wildlife in Mississippi along the coast, the Gulf Coast, and new snail. The zebra crayfish was just described from the Locust Fork of the Black Warrior, I think last year. A couple cave crayfish. This is probably the longest common name that there is. Sweet Home Alabama cave crayfish was described about 10 years ago now. Moving on, I think we've got another. And some of these things were just really discovered by looking underneath huge boulders in Shoal Creek of the Tennessee and Northwest Alabama. Found the bottle brush crayfish, which is about the size of a lobster. And the Lacan exit crayfish was found only because the entry ramp to South I-65 cut through a hill and opened up a cave, and a graduate student climbed in there and found this. So we're finding stuff all the time. It's pretty cool. But it does make keeping up with up-to-date maps difficult.
Yeah, so our goals coming into this were to identify and really to quantify areas of high biodiversity and unique fauna within the Southeast, and then to feed that data into a prioritization so that we could plan conservation actions and both recruit and target additional funding. As I said, we were approached to start this work by the National Fish and Wildlife Foundation, who at the time was starting to think sort of more strategically about fish and aquatic community conservation in the Southeast, and wanted a little help in sort of making the case that money that right now is going to the Gulf or going to salmonid conservation in the West far outstrips the spending that's going on in the Southeast, and we should really be trying to recruit new investment in conservation in the Southeast. With that in mind, we should have an idea of where that funding need is most dire and what the set of priorities for management actions would need to be to conserve that biodiversity. They let us sort of draw our own boundaries for what we considered the Southeast, and being fish people, we started with the fish data, and Bernie came up with this 290 Huc 8 study area. I hope all of you are familiar with that Huc designation. It's basically the way the USGS addresses river segments in the country, and the more digits you have in your hydrologic unit code, that's the Huc abbreviation, the more digits you have, the smaller the area described by that number. So you start off with Huc 2s, and the entire Mississippi drainage, I guess, is a Huc 2. We're working at the 8-digit Huc level for our prioritization here, and I think an average 8-digit Huc in the Southeast is about 1,350 square miles. There's 290 of those within the boundaries that we picked, and basically that is east of the Mississippi, south of the Ohio. We didn't pick the Hucs that straddle the Ohio, so anything that only drains north into the Ohio, and then we cut off peninsular Florida, both because it's such a mess down there with non-native species, but also because the level of fish biodiversity really drops off as you move south into the flat part of Florida.
And an example, the St. Johns River there, I think there's 18 species of fishes found along the Atlantic slope that stop in the St. Johns. So I tried to use logical breaks like that.
Yep. Our general procedure was to get point data of species occurrence, either from online databases such as the GBIF, the Global Biodiversity Information Facility, which is really just a meta catalog of museum data. We also went to the state natural heritage programs and asked them for their periodic monitoring survey data. We got a fair amount of data from individual researchers. When we got point data, we aggregated that to the Huc 8 level. So if a fish occurs anywhere within the, let's say, Upper Oconee River Basin, then the whole Upper Oconee Basin lights up for that species. Oftentimes the data were already aggregated at a finer scale because whenever you're dealing with threatened and endangered species, the natural resources agencies are very careful not to give people too much information about where those are found. There are people that will go out and seine up an entire population of a threatened minnow or a threatened sunfish or something like that for a hobbyist collection or something like that. And so they'll often obscure those sample locations a little bit, but that was fine for our purposes because we were then generalizing that data further to the Huc 8 level. We used all of those points to generate range maps for each species. Then we began the laborious process of checking those range maps against published literature and also convened some committees of people who could give us expert judgment in the absence of published range maps. And then from that, we were able to overlay those maps and calculate species richness, which is just a count of the species that occur within a watershed. Also endemism, which was a score that we derived, which is basically the inverse of rarity. If a species entire range is contained within one Huc 8, it got an endemism score of 1. If its entire range was in 4 of our Huc 8s here in the Southeast, then its endemism score was 0.25, and so on. We only considered something endemic if its entire range was caught by that 290 Huc footprint that we chose as the Southeast. And then we also calculated scores for imperilment, and imperilment here does not mean federal designation as a threatened or endangered species. Instead, we went to the professional societies that specialize in fishes, in mussels, and in crayfishes, and used the periodic summaries that those societies create and publish as a sort of estimate of imperilment that's not biased by the vagaries of the federal listing process. So we started our analysis with just over 500,000 records for fish locations, along with some polygon data. We, as I said, had to vet the resulting ranges. Bernie did all this using published Fishes of books for most of the states in the Southeast. There's some online atlases, including Georgia's. We only delineated native ranges for fishes, so we kicked out any species that are not native to the Southeast, and we also kicked out any areas where fishes have been moved around, like maybe flathead catfish, which are originally a Gulf species, but have been translocated by anglers all up and down the Atlantic coast. So we deleted those non-native distributions to the extent that they're known. And then, as we said before, we had to deal with new taxonomy. You want to walk people through this slide, Bernie?
Yeah, so museum records were obviously the basis for the vast majority of these data for fishes and mussels and crayfishes. And collections managers do not keep up with taxonomic changes. I mean, it may be decades before the names are changed. So here's an example. Ethiostomus stigmaeum used to cover the entire range of this of the map, as you see on the bigger map below the speckled darter there. And over the last 10, 15 years, it's been broken up into president's darters, and even before that into some other darters in the upper Tennessee. And so now we have a total of 10 different species. But some museums, it's all Ethiastoma stigmaeum. So you've got to actually address that and change those names in the database based on where the localities are, which is pretty laborious, but it gives you a much better, accurate picture of where the biodiversity is.
So we went from about 500,000 points of fish data down to about 40,000 points of crayfish data. However, we feel pretty good about using this crayfish data because oftentimes we were talking to the people who had actually collected the samples. The crayfish community is small and dedicated, and so I have sort of more confidence in any particular identification here because it's often a person with a PhD who is out there specifically looking for that particular crayfish that is generating these points, whereas the things that wind up in the fish databases could be somebody's ichthyology class out on a sampling field trip, or a team of summer hires working for a state agency that are trained but not necessarily trained at the PhD level on their fish identifications. And mussel sample distribution sort of sits in between those two. We have a lot more polygon data for mussels, and it tends to cluster in Alabama and the Chattahoochee drainage in Georgia. Just because that's where mussel diversity is highest, that's where people tend to do their surveys. Mussel taxonomy is not quite as fluid as crayfish taxonomy, but in our second revision of this strategy, we did wind up doing a fair amount of revision in our original mussel maps to reflect updated taxonomy and what our committee deemed to be misidentifications and that sort of thing in museum records. So as I said, we put together a committee of mussel experts there on the left, crayfish experts there on the right, although I'm in both of those pictures and I would not number myself in either group. Basically sat them down for 2 straight days looking over these maps. They would go home and pore over reference material and then come back and update our maps sort of interactively. And so that allowed us to generate scores for the Hucs. And as I said, richness was just a sum of species with records in a particular Huc. Endemism is a measure of how unique that species is to that area. And imperilment, we used a weighted imperilment, which gave triple points for an endangered status, double points for a threatened status, and 1 for vulnerable status. And we summed each of those scores. Just quickly, I can take you through the maps and see that for the next set of maps, there will be fishes, mussels, and crayfish insets on the right. You can see that they vary a little bit, although fish and mussels tend to track each other fairly well, perhaps because of mussels' unique life cycle, where they use fish hosts to distribute their larvae. We assume that that's a highly co-evolved relationship, and so there are reasons that fishes and mussels would probably track each other pretty well on a biogeographic basis. Crayfish have the complication that they get out and they walk around, and so they can obscure some of the watershed boundaries, particularly in the flat parts of the Southeast down on the Gulf coastal plain. But anyway, richness for all 3 species together tends to cluster in the middle and lower Tennessee, in the Green River system in Kentucky, and in the sort of eastern arm of the Alabama Mobile system. You can see for overall species richness, we've got hotspots in the lower Alabama and the Cahaba down there in the middle of Alabama. And feel free to jump in anytime if you've got stuff things that you want to point out, Bernie.
You got it right there. Keep going with Duncan.
Okay, endemism is a little patchier. We see fairly high endemism up in the Clinch River, right where it crosses over from Virginia into Tennessee. That's driven in large part by both fishes and mussels. That's not a real hotspot for crayfishes. Crayfish, the locus of endemisms, appears a little bit further west. And we see a lot of contribution from not only the Middle Tennessee, but also the Mobile Tom Bigby system has a bunch of orange hucks in there. But when you lay it all together, you're still seeing mostly the Middle Tennessee, a lot of the Alabama and Mobile system, and then a fairly good chunk of the Green River system in Western Kentucky and Northern Tennessee showing up.
And a little bit of the Cumberland.
And a little bit of the Cumberland, sure. And then imperilment, again, the Clinch up in Tennessee and Virginia comes up hot. That is both because it's fairly diverse, but also because of mining impacts, we think, in that area. Coal mines do a number on mussels, although there could be other things that are impacting that community there. And we have, again, a real hot spot for imperilment in the Pickwick and Wheeler Lake, that little arc in western Alabama that gets, in northern Alabama that gets over just into the northeast corner of Mississippi. We refer to those as Pickwick Lake and Wheeler Lake because that's what the USGS calls them, but those are really Tennessee River mainstem hucks. And we were surprised to see both diversity and imperilment come out so high there. But it turns out that, like, in the main stem of the Tennessee is not really where the critters are in those watersheds. That is a very karsty area. There's a lot of limestone in the surficial geology, which leads to a lot of springs, a lot of caves. And when you have headwater systems with a lot of cave and spring habitat, you can get a lot of very small distributions of very specialized organisms. And when you have small distributions and a high degree of specialization, you are a ripe candidate for imperilment. So there are a lot of spring and cave species that are causing those two hucks to light up. We decided, based on a fair amount of analysis, that richness, endemism, and imperilment, while there were correlations, obviously, in all of those maps, they were each worth keeping because they captured slightly different aspects of the biodiversity here. And our advisory committee decided that any mathematical processing we did on that was putting our thumb on the scales, no matter how explicit we were about those decisions. And so the best scheme was just to standardize those scores and sum them. But we acknowledge that that's itself a weighting scheme, and that other defensible ways of combining this information exist. But overall, take-home message for the Southeast is when we developed a priority sum, maximum score was 3. Wheeler Lake and Pickwick Lake come out 1 and 2, and they're almost tied. Wheeler Lake has 220 species total. It's 9th ranked for fishes. It's 1st ranked for crayfish. It's 2nd ranked for mussels. Pickwick Lake has 245 species total. It's number 1 ranked for fishes, number 2 for crayfish, number 1 for mussels. Follow that up with the Upper Clinch, which I've talked about a couple times already. The Lower Duck, which coming in, I would have thought was likely to be number 1. Historically, that's been the place that people go to see the maximum number of fish species. Follow that up with the Cahaba in central Alabama, the Caney, which is a Cumberland River drainage, Upper Cumberland, Lake Cumberland, again, a reservoir huck, not really a lake. Then the Middle Coosa and the Etowah and the South Fork of the Cumberland round out our top 10. one thing that I would observe here is that Wheeler, Pickwick, the Upper Clinch, and the Lower Duck are all drainages within the Greater Tennessee River system. 3 of the remaining 6 are drainages within that Alabama-Mobile system. And the other 3 are Cumberland drainages, which really tells you a lot about where we think the priorities are in the Southeast. Now, that doesn't mean that there's no reason to devote attention to watersheds elsewhere in the Southeast, because there's 220 species in these in Wheeler and Pickwick, but a lot of them are shared, right? And there are plenty of unique species that are not found in these drainages. But on a first cut, we thought that the middle and lower Tennessee, the Mobile and Coosa systems in that lower lobe, and then the Cumberland and Green were really where we would put our initial investigations for effective management actions for conserving biodiversity in the Southeast. Is that a fair way to say it, Bernie?
Yeah, I believe so. And realizing that the poor Atlantic slope. Let's not cry over that. Because if you do have money that is appropriated for that part of the country, we can reanalyze this data just for the Atlantic slope. We can reanalyze this data for South Carolina and really come up with priorities within any defined geographic area you wish. So that I think the data can be used many different ways, not just the top 10 throughout the entire Southeast.
Great. And of course, we can do this on the basis of fishes or mussels or crayfishes individually. So based on an early top 10 that we had, sort of the first time we went through this, we had some folks dig into the state wildlife action plans and other watershed planning documents. All I'll say about this is that we classified things into primary and secondary threats, and of course, urbanization and ag come out almost everywhere as being threats. But I'll also note that of particular interest to this freshwater flows group is that impoundments and barriers are at least a secondary threat for every one of our high priority watersheds. So what do we do with this information? Well, before we can prioritize new conservation, it would certainly be nice to know what the picture for existing conservation projects, sort of a capacity analysis looks like for the Southeast. And 3 years ago, we thought, oh, this will be easy, but the data was, shall we say, fragmentary. I started off by looking in the EPA Surf Your Watershed database to see if I could find where groups were and where they were focused. And there were, I think, 632 groups in that EPA database. And when I surveyed them, we only heard back from 51. And of that 51, all but 9, so 42 of the 51, had updated either their officers or their address or their area of focus. Fully half of the groups had updated their area of focus since they entered their information in the EPA database, which meant that we felt that the picture of groups winking in and out and changing their focus in the Southeast was such that we couldn't really analyze it using that unmaintained database that the EPA had up on their website. And I was very pleased 6 months ago or so to see that River Network is picking up where the EPA left off and trying to come up with a more curated picture of where people are working in the Southeast. I see great potential in keeping an eye in maintaining this data set and starting to overlay that with the sort of biodiversity priorities like we have developed. And here in our group, we have, in the group of people that developed the strategy, but up at the Tennessee Aquarium, they're using these priorities to design an information management system. You want to talk about this, Bernie?
Yeah, so obviously the Cumberland, Tennessee, and Mobile Basin were the hot spots, and so we decided, okay, since we know fish is better than any of these groups, and they're a little more charismatic than, especially mussels. You know, come on, they're rocks with snot in them, but no, they're pretty cool. But anyway, so we decided to go deeper into here and actually look at point data. So um, I'd cleaned up some of this point data quite a bit, but now rather than just having it in Huc 8s, I had to be more specific to the point data, and I'm still correcting these data. But so we're going to have an interactive web page where, first of all, there'll be museum data plotted. Okay, you can go to the next slide, Duncan. And then you'll be able to navigate this by family, by species. You can enter your address on this web page and find out the fishes in a certain area around you, fishes by a state. There's going to be a lot of ways for the public and scientists to use this website. But then what's really unique about it, next one, is it's going to be a living museum. So, you know, I work with over 20 species of threatened and endangered fishes, and they are no longer put into jars and put in museums. The only record of them are in people's the only records are in people's field notes or Fish and Wildlife reports. Well, I take photo vouchers of all my fishes, and so what we're doing now is with encouraging everyone to take photo vouchers, scientists, and eventually the public, submit them to us. We vet that they are the correct species, and then a dot goes on the map with a photo voucher as that quote-unquote specimen in a jar, but now it's a photo. And so we think this is going to be a much better way for scientists to be able to communicate with each other on species they're working with more updated information, and to get the public involved eventually. So we're hoping to roll this out this fall and have this ready to go. But that's just one example of how this analysis sort of said, okay, let's dive deeper into these three drainages and these 400 species of fish in the Tennessee, Cumberland, Mobile Basin. All right.
So there's too many people to thank individually, but this is all the folks that contributed data, or all the people that sat on our committees. We had both a crayfish and a mussel committee, but also sort of an overall steering committee. With that, I think we can take questions. I think April has a couple more survey questions that she can put to the group. I will say that the first version of this prioritization is still up at our project website at southeastfreshwater.org, along with an ArcGIS story map that kind of takes you through a lot of the same topics that I have covered on the talk today. I hope this has been interesting and worthwhile for you folks, and I'd be happy to clarify anything or expand on any topics you'd like to hear more about.
Yeah, please let us know what was asked.
Yeah, thank you so much, Bernie and Duncan, for leading this immense amount of work that you did, and also for sharing your work with us here today. I want to encourage everybody who is on the webinar to ask questions. There's a question box on the control panel. If you click the orange button with a right pointing arrow at the top right of your screen, you can type your questions in. And while we give you a few minutes to ask your questions, we have some questions for you. So I'm going to launch another poll. And what we'd like to know is, who else do you think needs to have this information shared with them? And you can select all that apply. Just so select all the ones that you think apply, and we will share what your thoughts are here in just a moment after we give everybody a chance to weigh in. And I will say that we're going to take this advice that you're giving us and think about how we can actually do what you suggest as well. So it looks like probably most everybody who's going to vote has voted. And so let me, looks like we have a few more people that are still weighing in. All right, I'm going to close the poll in 3, 2, 1, and I'm going to share the results. It looks like, let me see, we have about 75% of you think that we need to share it with conservation and environmental organizations, 75% with academic institutions, 67% with foundations and grant makers, 92% of you think this information needs to be shared with government agencies. So that is the top one that you all agree on. And then 67% of you say private sector, industry, and businesses. All right, I'm going to hide that poll. And then we want to ask you another question. Hold on just a moment. We want to know how you will use this information. So we've actually got 2 questions on that front. So how will you share this information with your networks, or will you share this information with your networks? And just please say if you know you will, if you know you won't, or if you're unsure. All right, it looks like probably we have most folks have voted that are going to. So let's close the poll and take a look at the results. So it looks like about 60% of you think that you will share this information with your networks, and about 40% of you are unsure. So that's great. Thank you for sharing that information. And the final question we have for you is what ways you might share this information or use this information, and you can select all that apply. So in your communications, in your funding or fundraising appeals, in your advocacy efforts, in your planning, either management planning, strategic planning, or maybe some other way you might use it that we didn't list here. We'll give you just a few more seconds to weigh in here. And it looks like we probably have most folks have given your input. So I'm going to close the poll and share it. And it looks like by far the most way that you anticipate using this would be through your planning efforts, but also with your communications and fundraising and advocacy. So that's great. That's great to hear. So what I want to do now is open it up and see what questions you have for Bernie and Duncan. So let me open up my questions here and let's see. So a question that we have is, how do you deal with fish who are being reintroduced to areas where they had historic presence but are no longer thriving? For example, river sturgeon in the Tennessee were largely gone from the reservoirs but are being reintroduced now. From my understanding, the reintroduced fish have not successfully spawned yet.
Okay, so I can answer that. So when we built this, if a species was extinct, we did not include it in our data set. If it was still extant somewhere, we included historical range because we can reintroduce it. And the example they stated, the lake sturgeon, the reason they have not reproduced yet, there's 2 of them. Well, we know they haven't because, one, the females take 20 to 33 years old before they spawn for the first time. Males 15 years, and this stocking has only been going on for, this will be the 18th year. So we probably have males that are sexually mature, females getting there. So we won't know for another couple years if spawning will occur. And then sturgeon need larval drift, and we don't know if the reservoirs have enough flow for the larvae to develop properly. So it's going to be another 5 years before we know. But you know, I think reintroduction has several different phases. There's reintroduction with continuous stocking, and then there's reintroduction with enough genetic diversity and life history requirements that it's a self-perpetuating population. And for lake sturgeon, that remains to be seen. But I think both types of restoration and reintroduction are valuable.
Great, thank you, Bernie. Duncan, do you have anything you want to add to that?
No, I mean, the Tanasi folks are intimately involved with all those sturgeon stocking operations, so Bernie's a much better person than I to weigh in on that.
Okay, we have another question, and I want to encourage all of you who are still with us to please type in your questions. We have time for a few more. and you can just type those right into the questions box. So the next question that we have is, what are the most impactful ways to use this information and data to either enact change or to get protective actions in place? What do y'all think?
Well, I think one of there's two problems in the Southeast with getting action done. One is the public, the electorate, the people that, put pressure on politicians to get stuff done. We don't have a very green public. This isn't going to help change that. But number two is the amount of money that comes into the Southeast. I've done analyses over the last 5 fiscal years that are available, money spent on federally threatened and endangered species in the United States. And for fish, on average, if you're a species found completely outside the Southeast, you get 46 times more funding than if you're a species of fish inside the Southeastern United States. The whole budget for the Southeastern United States yearly averages about $6 million. One salmon out west gets $270 million a year. So the numbers are just astounding. So if we can use this to highlight the biodiversity in the Southeast and then sort of hone into where this money should go to get the greatest bang for the buck with the limited resources we currently have, we can spend wisely and hopefully highlight what we have and get more money to the Southeast. But I think, it all comes back to the public. The public has to tell their representatives and senators, go to Washington and fight for conservation dollars, and that doesn't happen in the Southeast.
This is April, and Catherine Bear, my colleague at River Network, who is the director of science and policy at River Network. Catherine, I have your line unmuted as well if you want to weigh in here as well. But there's similar statistics in terms of private foundation dollars that are invested in environmental organizations, environmental NGOs in the Southeast as well.
Yeah, and that's a good point. When when Bernie was saying that, it made me think that there's that parallel there that of all the foundation dollars that go to environmental funding, April, I probably won't get the numbers exactly right, but of it, the entire was like 9 to 11%.
Yep.
All the southeastern states. So there's also some inequalities there for various reasons, and so they too may well be correlated.
Yeah, and the other thing that I would add is that Duncan and Bernie have put together a really great website, which a link to it will be in the follow-up email that you'll receive tomorrow, and I'll also paste it in the chat. But those ways that we just listed and ways that you might share this information in your communications, in your funding proposals, in your advocacy, or are all other ways to think about how you might want to share this information with those different audiences as well. Duncan, do you have anything you'd add?
I don't think so. I will say that this project has pretty good visibility within the fisheries program and the at-risk species program for this region of the Fish and Wildlife Service. I work with those folks frequently and have given them slides of these maps and that sort of thing for use in both internal and external communications. So we are trying to push this information out to not just folks who might give us money, but also folks who are doing a lot of the conservation work from the ESA side in order to influence decisions and hopefully help them make the case within their channels as well.
Great. So we have another question, and again, Catherine, if you want to weigh in here as well, I'm sure it'd be welcomed. So the question is, how critical is the waters of the U.S. rule to protecting freshwater biodiversity in the Southeast?
Yeah, I saw that, and so I think probably everyone on this call knows the Clean Water Rule is a hard-fought rule that was promulgated under the Obama administration and has been the Trump administration is trying to repeal and replace it with something that would be less protective of the scope of streams under the Clean Water Act. So I think in general, the rule is really important because it does a lot to protect some of our smaller streams and rivers and the adjacent wetlands to them from development. And, you know, it'd be interesting to see sort of the development pressure overlaid with the maps that you all showed, but certainly wherever we see that pressure and there's a move to, you know, development and pave over streams, that's when, you know, that would have a big impact on the smaller streams, but also the downstream waters that we're impacting that way. So I think to Sandra's question, it is critical to, you know, all of our streams and rivers throughout the Southeast, but definitely has a role to play in protecting the biodiversity we've seen here.
Yeah, and I will add to that so much of this diversity, as Duncan alluded to earlier, is not main stem. The main stems have been trashed by reservoirs, and it is in these smaller tributaries. And the specialists are quite often in wetlands and springs and caves that, you know, are sort of hidden waterways that, you know, are quite often manipulated and changed by humans. And so lessening those rules are really going to effectively direct the aquatic biodiversity. There's no doubt about it.
Sure. Anything you do, you know, if you're spring endemic and somebody changes the local water table by just, you know, 10 inches, that can greatly impact the amount of spawning habitat that you have at a certain, you know, at a particular time of the year. Or, you know, one of the most obvious aspects of that clean water rule had to do with whether you call them ephemeral streams or whether you call them ditches, you know, small unmapped creek segments on private land. And there are a number of species, you know, a number of the things that are driving those endemism calculations are using those creeks, or are impacted pretty rapidly by sedimentation that is delivered to somewhat larger creeks by those smaller creeks, and lessening controls on, you know, very small scale land use change can pretty quickly impact aquatic habitat for a critter that has limited range and is a headwater specialist.
Yeah, so summarizing what I think I heard all three of you say was that the efforts to repeal and replace the clean water rule will likely result in smaller headwater streams being removed from those Clean Water Act protections, and those protections are really critical to protecting the habitat of a lot of these species that make the Southeast so special in terms of its biodiversity.
Correct.
Yeah, that's my opinion.
All right. Well, it looks like we've answered everyone's questions thus far, and we are at the end of our time. So I want to quickly just bring up one final slide and to say farewell to everybody, and just a reminder that we did record today's webinar, so and that recording will be made available on our website, as well as a link will be emailed to all of you who are in attendance today. A follow-up survey is going to pop open once the webinar ends. Please take that survey. It really provides us with valuable feedback and makes a big difference. And want to say a really big thank you to Duncan and Bernie for not only doing this important, massive amount of work, but also for taking the time to prepare and share that information with all of us here today. Thank you so much. And thank you to all of you who attended the webinar today too, for all of the great work that you do to protect and restore your local waterways. And you are welcome to contact me. Again, I'm April Engel. You can reach me at aengel@rivernetwork.org, and I'd be glad to connect with any of you to answer any questions. And I hope all of you have a wonderful afternoon, and farewell. Bye-bye.
Bye-bye.
Bye-bye. Thanks again.